So I'd like to thank the organizers to give me the opportunity to talk about hyperuricemia and mechanism of what we see in hyperuricemia and how that can lead potentially to development of gout. So today I give you an overview of what hyperuricemia is, let's say, associated with a certain disease. And of course, you all know as rheumatologists that it's strongly associated with gout. But we know also there's association with metabolic syndrome, atherosclerosis, type 2 diabetes, non-alcohol fatty liver disease, chronic kidney disease, hypertension, but also, for example, with scleroderma, we see also an association with hyperuricemia and pulmonary hypertension. So hyperuricemia is linked to many, many diseases. So when you look in hyperuricemia per se, and this is the paper we hope to, actually we published in the last issue of iScience. There what we did, we have a large cohort collected here in Cluj of 250 roughly healthy people, 250 people with hyperuricemia, asymptomatic, and 250 gout patients. And we took serum, and from the serum we analyzed the inflammatory status with targeted proteomics. Nowadays you have a very fancy technique that you can do up to 100, 200, and even more proteins in 25 microliters of serum or plasma. And we ask ourselves, what does hyperuricemia per se does in the inflammatory status of the patients? And as we can see over here, you see over here that this is asymptomatic hyperuricemia, and you see all these orange dots are significantly upregulated inflammatory proteins. And what also we did hyperuricemic patients with gout and hyperuricemic patients without gout, and then you see that only a few markers still significant. So the majority of the inflammatory proteins in these patients are coming from hyperuricemia and not coming from gout. And of course, gout, as we know, is an inflammatory disease. And most of you probably studied that as an R1 disease. So what we did in the past, we measured in controls people with gout, so asymptomatic, intercritical gout, and during attack. And this is a method that we can measure very sensitive cytokines in the blood. So it's kind of based on ELISA. And what you can see already here, that in the gout, you can see this R1 is elevated, R6, R18, but VEGF as a control, there's no differences. But you see immediately, when you have a gout attack over here, you see a strong elevation of R1 beta. So also see that gout is a really inflammatory disease, and you can measure these inflammatory mediators in the circulation. But what happens if you look at cells? When we take cells from gout patients, PBMCs, and we stimulate it with a gout-related stimulus, in this case MSU crystals. And what you can see here in this slide, you can see the R1 beta production from PBMCs, from controls, healthy control of patient. And what you can see depends on the stimulus. The black bars, you see that gout patients produce much more R1 beta than the controls. And then we separate the patients. So this slide is only gout patients with low uric acid and high uric acid. And then you can see that patients with the highest concentrations of uric acid in the blood produce the highest levels of R1 beta, indicated as a link maybe between uric acid and the production of these pro-inflammatory cytokines. And then, of course, we say, okay, if this is uric acid, that uric acid drives that, can we mimic that in vitro in our lab? So what we did, we took cells from healthy volunteers, PBMCs or monocytes, we exposed them for 24 hours to a concentration of uric acid, and then we re-stimulate and measure the cytokines. And what you can see over here, you see R1 beta again, and you can see different concentration of uric acid, stimulation, and what you can see, the highest dose of pre-incubation with uric acid caused massive induction of cytokine R1 beta. But very interesting to see that when you look at the natural antagonist of R1, is R1 receptor antagonist, is downregulated. So we see a clear shift in higher production of R1 beta and lower production of the inhibitor. And we also could confirm that when we look at the mRNA over here for R1 beta, and this is R1 array is down with the uric acid, and here R1 beta is up, indicated that this really uric acid can mimic this in vitro. So then, of course, we use these fancy techniques like RNA sequencing. We expose monocytes to uric acid, and we look which genes are really up and down regulated. And what this slide shows you is very easy. You can see this. We expose the uric acid, re-stimulate now with LPS. You can see there's a higher peak on the R1 beta gene over here. But most impressive is here. Look at the R1 receptor antagonist over here. You see that the uric acid, this is a control, and when we expose the monocytes, in this case to uric acid, there's almost no transcripts for R1 receptor antagonists. This could really confirm that. And then of course, we have all these genes. You can do pathway analysis to see which pathways are upregulated or downregulated. And again, what you can see here, there's R1 beta signal and pathway strongly upregulated when the cells are exposed to uric acid, soluble uric acid, and R1 receptor is downregulated over here. So indicated that urate per se can have an effect on the monocytes, so make them really pro-inflammatory. So how is that regulated? We know that these imprinting or changing in phenotype often happen with epigenetic modifications. So you have inhibitors to add to the cells to see which epigenetic mechanism play a role. And what we did in this slide, actually, the same experiments we took. In this case, PBMCs from healthy volunteers, we add them with uric acid, and then we add inhibitors. One inhibitor is an acyltransferase inhibitor or methyltransferase, and both enzymes are linked to epigenetic modification. And what we clearly see, that if we add this inhibitor over here, there's no effect actually on the training of uric acid. See over here, when we add a methyltransferase inhibitor, then we could really see the difference. You can see this is the control, this inhibitor, so the elevating cytokine production is down, indicating that probably this enzyme methyltransferase has an effect on the uric acid-induced higher cytokine production. So how can we explain this? And this is a new concept that we introduced already 10 years ago, that not only T's and B cells have a memory that they encounter a certain pathogen or certain protein or antigen, but also cells from the myeloid cells, monocytes and macrophages have a kind of a memory. It's not a classical memory as you see in T and B cells, but this memory is built on a first insult. So when monocytes have a first insult, for example urate, they produce a certain amount of monocyte, but after waiting for several days, and you stimulate the cells again with the second stimulus, these make much more inflammatory. And it has to do with these modifications on the histones. And that's what we called epigenetic modifications. So what is, when you look more carefully, what happens then, and this you see this is the first insult over here. We have, for example, we have data on vaccinations, but also exposed to fungal components, but also oxal-LDL in case of cardiovascular disease, and we add now hyperuricemia to it. For the first insult. But after the resting period, then you stimulate the cells again, you see much more pro-inflammatory cytokines. So what happens then, during this period here, this methylation happens on the histone tails over here, promoters much more active, and then you have much more mRNA and more cytokines. And this is in the resting state, you see less immune genes. And we know that there can be these histone modification over here, but also now we know that these long non-coding RNAs, they're also important to add these histone modification on the promoters. So when you talk about epigenetic modification, you have actually 4 major categories. Today I focus only on one, but we know about DNA methylation. There's also long-term modification of the DNA, the methylation of the DNA. The microRNAs are important. Histone, this is long non-coding RNAs and histone modifications. And today I want a little focus on this one over here. So this is a scheme to explain what happens in this trained immunity induction or training or priming of these myeloid cells. So what we normally do in our laboratory, we isolate monocytes from healthy donors, we expose them to the first stimulus. Mostly we choose for urate exposure. We wait several days. For example, we wait 1 or 2 days for urate, and we can wait longer when we use oxal-LDL or heat-killed candida as a strong stimulus. And then in the second time, we re-stimulate with crystals, so gout-related stimuli. And what actually happens in the beginning, this is the situation in the histone tails over here, and you see here HK3 trimethylation over here. And then the resting period, and the resting period happens then you see there's a much more of these modification happens on the promoter. And after the second stimulation, then you see that much more mRNA is described and more cytokines. So this transformation from here to here, that's what we call epigenetic reprogram. So, of course, we're very interested to study that. And there's an assay and a technique you can measure to all genes linked to this particular histone mark. So what we did, we exposed human monocytes again to urate, and then we look at the histone modification. And seeing the data coming from the other stimuli, we will focus on two major marks. That's the histone mark over here, that's the H3K4 trimethylation, and the 27 acetylation. So what you do with chip sequencing, you have antibodies against these histone tails, you precipitate with the DNA connected to them, and then you sequence all the DNA, see what's linked to this mark. And that you see over here. These are the results from this mark, from the K4 trimethylation and this K27 acetylation. And to our surprise, we found nothing, no signal at all. And of course, it was a little bit disappointing because we showed that it's important, we have the inhibitors, but probably what we choose too fast. Now we know that other histone marks are also important for different stimuli. And probably for urate, we have no indication that we look at the wrong mark. We have to look at this one, H3K9. There's another lysine on the histones, and that is modified. So we're working now on this particular mark to see whether we see genes linked to this modification, the urate exposure, and can explain why these cells make more cytokines, IL-1, IL-6, and less inhibitor like IL-1RA. So this one part I want to show, a new mechanism. Of course, since that we saw this form of training that we say, okay, the monocytes are modified during exposure to urate, of course the big question is then, okay, are all the monocytes equally affected by urate, or are certain subtypes more sensible to uric acid training or priming? And therefore now there's a new technology available called single-cell RNA-seq. What you do then, you isolate one cell, and then you send the isolated RNA from that cell and sequencing, and you can see exactly which cell produce certain genes or transcription. And that's what we use. And you can see the data over here. This is from gout patients, 8 gout patients and 6 healthy controls. And this works as follows. You isolate PBMCs, and each cell is in a droplet system. It's analyzed. We isolate the RNA sequencing, and then you can look at the total transcriptome per cell. And what you generally get is these type of pictures. You can see the cells. For example, here you see the monocytes, CD14 positive monocytes over here, and here you can see CD4 T cells. But you can separate the cells very nicely on the transcripts on an RNA sequence pattern. So we were very interesting. Okay, what's happening here? And you can see again, you see here the major cell population that we're interested in is the CD4 monocytes, because we're interested in epigenetic reprogramming of the monocytes, but you can see also the T cells. And then we look which genes, the DEGs, oh sorry, I have to go back. So the DEGs stand for different expressed genes. You see that in the T cells, in the gout, we have more upregulated than in the healthy controls. And also in the CD14 monocytes, you see that in the gout patients, we have more transcripts than in the controls. But also for other cells, NK cells, for example, we see differences. But now we focus on the monocytes. So what we did then, we analyzed these monocytes and look which genes are different between the controls. And we found 22 genes that were clearly different between gout and healthy control. And one gene we're very interested in is this one over here, because this is a transcription factor, NA4A1. But the other interesting gene, but this took us our intention, because this is a gene that's linked to the phenotype of the monocytes. They keep the monocyte in its kind of M1 status, so a more pro-inflammatory status. So it's very interesting to see how this gene is regulated. And then we analyze it more in detail to look very specific on this gene, and you can see it over here. This is a gene over here, the NA4A1, and you can see that in gout patients, there's almost no transcript left. And the other is also down, but this was really downregulated, completely downregulated. And it's also known as a negative regulator of NF-kappa B. So it's an inhibitor of NF-kappa B signal. And then you can understand if this transcription factor is downregulated, that's why normally these cells exposed to urate or in gout patients have a more pro-inflammatory status. And also what I mentioned, it's very strongly associated with the anti-inflammatory type of the macrophages. So what we did some experiments to see, can we confirm that? So what we did again, we have cells from healthy volunteers. We expose them to urate, and then we stimulate them with LPS in this case. And what you can see over here, we have A donors. You see that we, after this is the R1 beta production, you see before and after uric acid exposure, you see that many goes up and few are more or less stable. And then we look with PCR whether this gene was downregulated or not. And what you can see over here, this is the gene NA4A. You can see in the responders, we should clearly see downregulation over here. And in the non-responders, there was no effect. So the persons individually saw no elevated R1 production. They have no difference in this gene, only those here over here. So they give an indication that it's probably linked to R1 production. So we came to the following hypothesis. So it could be that in gout patients or in hyperuricemic, asymptomatic hyperuricemia patients, that this factor is downregulated, this gene over here is downregulated, and then the cells are mostly in a type 1, more pro-inflammatory monocytes or macrophages. And due to hyperuricemia, this is downregulated. And normally you have a balance between the classical monocytes or the non-classical, more anti-inflammatory monocytes. So it means if this is down, the R1 beta is up, R1RA is down, and in this state it's the opposite. So of course it's a very nice hypothesis, and we tried that in vitro, it works very nice. But as we all know, in vitro experiments, maybe it's not correct. We want to see what happens in patients, what we see. And therefore we performed the following study. We called the discontinuation urate lowering trial. So what we did, together with the clinic in Nijmegen, we asked 35 gout patients who were well controlled for many time, but they were willing to stop the allopurinol treatment to see what's happening after a couple of months, a year with the gout and gout attacks. Are they under control? So this is the hypothesis, what we say, what could happen. So this is at the start of the trial. The urate is still low, and after 2 weeks or at several time points, we take several points, but the uric acid should go up. And also the serum levels over here. And we expect that the R1 beta production is going up as well, very fast. And then you see that the gene N4A is downregulated because of the increasing serum urate levels and concentration. So that's what we expected and predicted. Now let's see what happens then. So this trial, we started almost 1.5 years ago, and we almost finished the trial. We are waiting now for the last samples at 2 years time point. And what we have in this trial, we have, of course, we look at the genome, transcriptome, proteomics, I will show some data, but also look at the cytokine production, what happens with the PBMCs. We look at the phenotypes, the monocytes, but also we have some crystals. In the beginning, we did a scan to see whether the crystals are there, yes or no, or whether crystals are accumulated as you expected. If the uric acid goes up for 2 years, you expect more crystal formation. So we hope to have also the data on that, but that's later. Yeah? So this is the first results on the serum urate. And then as expected, if you stop the allopurinol, you see in 2 weeks, all the patients go rapidly up. And that's, of course, what you expected if you take the medication away. And you can see the lines over here, but you see the individual dots over here. You can even see better. These are the 12 months. Now we have data for 1.5 years and already also for 2 years, and they stay up. They stay really up, so there's no decline, whatever. So it means that allopurinol works. that's what we conclude from this. But of course, this we know, we expect it. But the next is most important. What happens to these monocytes? Do we see the shift in monocytes, yes or no? And what you can see over here, this is the type 1, the classical monocytes, and you see kind of drop here, what you did not expect, and then it declined. You see after 12 months, there's more than in the beginning. I mean, look at the non-classical one. We don't see a really difference between the time point when we started and the 1 year point, but you see clear when this is really low here, this is high, you see, and then it goes down. So something happens as well there, but we have to wait until we have 18 months and 24 months to see what's happening on the subset of monocytes. But the most interesting is over here. When we took out the PBMCs, the cells from the patients, and we stimulated in the lab with crystals and LPS to see what's the R1 production, what's the cytokine production. But you can clearly see here that in the first 3 months, nothing happens. And we saw that after 2 weeks, the uric acid is already high. So it has a delay. And what's really surprised to see after 6, but especially 9 and 12 months, see, the cytokine production is almost doubled. And we have already the first data from the 18 months time period was even higher. So it seems to be there's a delay. The monocytes are really becoming pro-inflammatory. And we see also what we saw with the IL-1 array over here, that the IL-1 array production slowly are going down, 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 down. See? So in what we saw in vitro, in our lab, with the cells, we could really see happens in patients. Although it took 6 months to 9 months to see this. And why that is, we don't know, but it's very interesting to figure out why it takes so long to have these phenotypical changes. Of course, we look also at the proteome, as the first slide I showed you, the same technology. And what you can see is also very interesting. You see in 2 weeks' time, this baseline, so here, before we stop the medication, and 2 weeks, no difference at all. Look here at 6 months, nothing at all. But look what happens after 12 months. You see, in the circulation of these people who have still no gout attack, they have hyperuricemia. Again, you see many of these inflammatory mediators are popping up in the circulation of these patients, indicating that elevated uric acid is really detrimental for the patients. Because we measure almost 100 different proteins and we can separate it per category. And what you can see, for example, MMP is a metalloproteinase. You can see after 12 months is really upregulated. Also, there's a VEGF is up, but also for intercellular proteins, you see they all go up. And also for membrane proteins, we took, for example, PD-L1. You see PD-L1 is also way up after 12 months. In these patients, in the serum of the patients, we can measure that. So, for conclusions, what I try to explain that the mechanisms how hyperuricemia can lead to inflammation. Whether it's gout, maybe contribute to other diseases like type 2 diabetes or hypertension. We don't know yet, but we focus now on gout. And what we clearly demonstrated, hyperuricemia per se results in systemic inflammation. We see it in the serum of these individuals. We see that the monocytes have an enhanced inflammatory status, they produce more. We also show there's a long-term effect. So the monocytes have long-term, let's say, changes based on trained immunity or epigenetic modifications. We also saw that the transcriptome clearly saw that IL-1 beta signals up and the IL-1 array is down. We found epigenetic modifications, as this probably links on H3K9 modifications. And we identified this gene as an important transcription factor in gout. And also, what for me was the most striking result so far in the STOP trial, that it took 9 months after hyperuricemia before we could see it in humans. So it means it's very slowly progressing, driving these monocytes. So it means that it has to do something with the progenitor cells. So it happens probably in the bone marrow, because otherwise I cannot understand why it takes so long. So what we're doing now at the moment, we are really interested in what happens in the bone marrow and the stem cells of these people. Of course, bone marrow aspiration is not easy to ask. Therefore, we're developing now technology to have stem cells from circulating blood to see if we can isolate them and explore them. What is the status of that? So in this kind of summary, what I try to show you today, that you have normally, if you have a normal uric acid concentration about this, nothing happened. As soon as start of hyperuricemia, of course we know the crystal formation, crystal disposition in the joint, and later on can lead to trigger gout attacks. But also now we know that has systemic inflammation, and we see that in the circulation all kind of inflammatory mediators pops up. We know that uric acid also has an effect on the monocytes, so it can cause strained immunity through epigenetic modification. And then when the second trigger comes, it can be an infection, or no trauma. These monocytes produce much more cytokines and can cause acute gout flare or even chronic gout and tophus gout deposits. And these strained monocytes, we know they have epigenetic modifications. They respond very fast, for example, to food changes, alcohol, fasting, but also crystals, as I mentioned, infection. And then the inflammasome is already active, and then we know R1 beta is produced, and that's caused gout. Because gout, again, is an R1 disease. Yeah? So what we want to do in the future, of course, we are very interested in these long non-coding RNAs, how they regulate these long-term modifications. How does it work? So we at the moment we're doing experiments in the gout patients versus hyperuricemia. Therefore, we're using the cohorts that we built, also includes here, together with the Department of Medical Genetics and the Rheumatology Department, together with Professor Rednick. We're working on the HINT-1 project. Now we got a new HINT project, HINT-2 project. But also we have a cohort in the Netherlands. We have an obesity cohort that we have majority of hyperuricemia. Only few patients have gout, but they mostly have hypertension, obesity, and these kind. And we also want to study in that, how is that linked to comorbidities. And of course, we were really, I think, the epigenomics, if we identify how it's regulated, it can give us new targets, not only in gout, but also hyperuricemia-mediated other diseases and comorbidities. And as last, of course, I want to thank all the collaborators, because this is work for many groups. First of all, the group in Nijmegen, we were help us with the studies, but also here in Romania, with a lot of help with our PhDs and postdocs, to collect all the samples from the patients and do all the analysis. And a lot of people help us with the fancy technology, for example, single cell, RNA-seq, long non-coding, but also people advise us what type of patients we have started also nice collaboration with our colleagues in Paris to get more information about the different forms of gout patients into critical for acute, and also very interesting in tophus gout. Do we see similar effects there, or is more severe training because they have huge tophus deposition? Okay, I think that was my last slide. I will thank you for your attention.